There we
Okay.
go. And I'd like to introduce Dr. Glenn Morrison. Dr. Morrison is the Vice President of Clinical Development, Neuroscience, and Rare Disease at Recursion, where the sponsor of the REC-994 Sycamore trial. He has worked in the rare disease and central nervous system space for many years, and we are very fortunate to have him here tonight to share updates. Dr. Morrison, if you'd like to get started, we're all ears.
Absolutely. Thank you, Connie. Let me start sharing. And all right, excellent. Well, again, thank you, Connie. As Connie mentioned, my name is Glenn Morrison. I am the VP of Clinical Development at Recursion. But more importantly, I am the program team lead for the REC-994 program. And on behalf of Recursion and the team, I'm very happy to be here today to speak about Recursion's ongoing efforts in CCM, and specifically about the phase 2 Sycamore study. So you can see the agenda here. I know many of you have had the opportunity to listen to the webinar that was presented last year, and at that time, the Sycamore study was not yet open for recruitment, so not too many details about the study could be shared. What I'd like to do today, in addition to introducing you or reintroducing you to Recursion and the clinical trials process, just a couple slides there, is really spend most of my time giving you an overview and an update on the Sycamore study. So with that, Recursion is a tech-enabled biotech or biopharma company. We are located and headquartered in Salt Lake City, Utah. We were founded in 2013 and we went public in 2021. And we are what is known as a clinical stage company, which means that we have clinical studies ongoing in which we're testing the safety and efficacy of our medicines in study participants. So this slide here is a cartoon meant to show how the special sauce, so to speak, of Recursion and how compounds progress from the lab to the clinic. Recursion has a bold mission, and that is to decode biology to radically improve patients' lives. And to do this, we are leveraging technology, biology, and chemistry, and medicine to build what we call the Recursion operating system. And our goal with the Recursion operating system is to build a biological and chemical map or series of maps, which we call the Atlas, and then to follow the Atlas by generating and following the data to identify potential medicines with a high chance of success. And then those compounds will move to the clinic either through partnerships or through our in-house clinical development activities. Now, from a tech-enabled drug development space, Recursion really is heads and shoulders leading the way. We have a dedicated team of over 400 scientists that conduct 2 million or more experiments per week. And this has led to the need for Recursion to really have an awesome computing power. And to that end, we have one of the top 100 supercomputers in the world with probably now more than 13 petabytes of data. And if you're wondering how much data that is, the way our CEO likes to describe it is if you take every feature-length movie that has ever been made in high definition, you still wouldn't fill all the available space on our supercomputer. So it really is some massive computing power. And we are leveraging all that data to build an internal pipeline, which currently has 4 drugs in the clinic, with another 10 or so that are close to being in the clinic. We also have partnerships, notably the Neuroscience partnership that we announced last year with Roche, and also the recommitment and partnership that we have ongoing with Bayer. But what does all this really mean? Well, we're reimagining the way that pharmaceutical drug development works. And what this means is that we want to fail early. Only the compounds that really have a high probability of success move forward through the testing process and then to clinical trials. And this really is meant to allow us to lower the cost of drug development, move faster to the clinic because the time, money, and people power are focused on those high probability targets. And so when we look at the drug development life cycle, which is shown on this slide, from target identification on the left to the ultimate goal of FDA approval on the right, our process can help us move faster by either eliminating certain steps, and this is shown in the top for what's known as known chemical entities or repurposing of drugs that are already approved, or we can expedite steps, which is along the bottom for what's called new chemical entities, or essentially newly identified compounds. And this really has panned out. So I'm showing here this mushroom figure, and it has the various disease areas that we're looking at along the bottom, from neuroscience to cancer to rare disease to inflammation and immunology, and along the right-hand side where they are in their life cycle. And what you can see, looking at the two pinkish mushrooms towards the middle, is that we have 4 compounds that are at the top stage or in the clinical phase. And notably, the one that's outlined in green here is REC-994 for cerebral cavernous malformation. So this really does set us apart from other tech-enabled biotech companies, and really very strong indicators that we will be able to deliver on our very bold mission. So let's turn our focus now to CCM. And I know you are all experts, and so I'm not here to educate you really, but just to set the stage for the rest of the discussion. And with that, just want to review a few points and agree with you on a few points. So CCM is classified as a hemorrhagic stroke disease, and that's because the abnormal collection of blood vessels, which can occur in any part of the brain, can bleed. CCM can be either familial, caused by mutations in 3 node genes, CCM1, CCM2, and CCM3 for simplicity, or it can arise sporadically. And cerebral cavernomas can occur in individuals of any age, size, race, or gender. And the symptoms can affect a whole host of body systems, from seizures, headache, memory issues, stroke, as we mentioned, to hearing loss, balance issues, tremors, and unfortunately, in some cases, even death. And as of today, there are no drugs specifically approved for CCM, and surgery, when it is feasible, is the current standard of care. Now, getting a little bit more technical, what I want to show on this slide is the mechanism of action of REC-994. And so healthy cells that line the blood vessels, and shown here on the left-hand side, through a series of interactions, release a compound called nitric oxide. And nitric oxide helps to maintain the integrity of cells. Mutations in the CCM genes, shown in the middle bar, and here we're looking specifically at CCM2, but it applies to CCM1 and CCM3, it leads to loss of CCM proteins, disrupting these interactions and resulting in, amongst other things, deficits in releasing nitric oxide and a buildup of reactive oxygen species. And this in turn leads to increased leakiness of blood vessels, localized lesion formation, and bleeds into the brain. And this oxidative stress, as it is called, can also lead to downstream secondary damage and additional mutations. Now, REC-994 is a reactive oxygen species scavenger that works to reduce the oxidative stress. And so in cells treated with REC-994, and this is shown on the right-hand side, the underlying mutation is still evident, but the buildup of nitric oxide is mitigated, so now it's released from the cells. And what we see in animal models is that this restoration of nitric oxide signaling really leads to the reversal of the cell leakiness and to a decrease in the number of CCM lesions. And what this looks like in the scientific data is shown on this slide. And so on the left-hand side, showing an indicator of leakiness, where higher is worse, is leakiness of cells from a control mouse treated with placebo in black and treated with REC-994 in white. And this is the OSM-WT label. And you can see with placebo or with REC-994, the leakiness is about the same. When you look at the 2 bars beside that, these are cells from a mutant mouse that has CCM mutations. And on the right, you can see that placebo in black. So you can see a massive increase in the amount of leakiness. It goes way up. And when they're treated with REC-994, the leakiness returns to normal, as you can see in that white bar. The middle figure shows another similar presentation, but this time with looking at the effect of blood vessels' ability to dilate. In mutant mice, this is compromised, as seen by that lower black line. And with REC-994, it's restored to that of normal mice. And you can see that's the top two lines that are together. And then on the right, we ran an experiment where we fed food with REC-994 or to mice, or we fed them a standard diet without REC-994. And mice on the standard diet developed a number of CCM lesions shown in black, and animals fed the 994 diet had fewer lesions as shown in white. And so it's really this collection of data that has provided the bulk of the information that we provided to the FDA, the US Food and Drug Administration, to gain approval to allow us to investigate REC-994 in people in clinical trials. Just a word or two on the clinical trial process, just to orient everybody. Typically, there are two stages that we like to refer to, the learning stage and the confirming stage. And these include three main phases of clinical trials before approval. First, we have the learning stage. This includes what we call phase 1 studies. Phase 1 studies typically, not always, but typically are conducted in healthy adult volunteers who do not have the disease. And we're looking at studies here to establish an early safety profile for a single dose and then for multiple doses, and learn about pharmacokinetics, or what the body does to the drug, and also about pharmacodynamics, or what the drug does to the body. And from there, we move to phase 2. We're still in the learning phase here, or learning stage. This is typically the first opportunity to test the safety and efficacy of the study drug in people with the disease or disorder. These are usually small focused studies that provide information for us, again, on safety and efficacy, but also help guide us to design larger studies in the next stage, which is a confirmatory stage. And this is our phase 3 studies. And typically this is in many more people with the disease or disorder. We're typically treating for longer periods of time, and we're looking to really prove that or confirm that the drug works as intended. And then these are the studies that we would then base our application for approval to the FDA and to other health authorities around the world on. I did want to show this slide because we do hear a lot about diversity, equity, and inclusion these days in the clinical trial process. And I just want to sort of frame this in the context of rare disease. Very importantly, people with a rare disease like CCM do represent a very important element in the conversation. Rare diseases, by their very nature, include people who are underrepresented in our healthcare system. And we need to do all that we can to run clinical studies for these rare diseases and bring promising treatments forward. And really the best way for us to do this is to partner with organizations like the one that Connie is leading, and to work with patients and their families like yourselves. And so we are advancing REC-994. This data is the phase 1 data. This was in healthy volunteers. We've conducted 2 phase 1 studies in adult volunteers, and we demonstrated that at single doses up to 400 milligrams and multiple doses up to 800 milligrams are safe and well tolerated. And in both of these studies, the safety, especially of the highest doses, really look very similar to that of placebo. And it's those phase 1 studies that really have set the stage for the Sycamore study, which is a phase 2, randomized, double-blind, placebo-controlled clinical trial to evaluate the safety, efficacy, and pharmacokinetics of REC-994 in the treatment of symptomatic cerebral cavernous malformation. So the study design is shown on this slide. We will screen about 80 people to identify and enroll 60 adults with symptomatic CCM. Eligible participants will then be randomized equally to receive either once daily doses of placebo or 1 of 2 doses of REC-994, 200 milligrams or 400 milligrams. Treatment will be 12 months with daily administration. And there will be 7 study visits over that course of those 12 months, really in an effort to try and reduce the overall burden. But we have visits on day 1 and day 2. And on these 2 days, we will be focusing on some blood samples to collect blood for the pharmacokinetic evaluation. Again, we want to see what what the body does to the drug. And then we have visits at month 1, and then every 3 months for the rest of the year. And at the end of that 12-month treatment period, we are starting an extension study where all participants will have an opportunity to continue into that study and receive REC-994. Participants who were receiving 200 milligrams will continue to receive 200 milligrams. Participants who are receiving 400 milligrams will continue to receive 400 milligrams. And those who are on placebo will be equally randomized to one of those 2 doses. But everybody in the extension study will have an opportunity to receive the active study drug. And as the title of the study suggests, we are hoping to get a better understanding of the safety and tolerability of REC-994, really to better understand the outcome assessments that will be most meaningful and most impactful, all with an eye on designing the phase 3 confirmatory studies. This slide outlines who is and who is not eligible for study participation. We really tried to make this as inclusive as possible. So eligible people for the study are adults 18 years of age or older with documented CCM lesions. Those lesions need to be related to symptoms, and that will be determined by the study doctor. We also have something called the modified Rankin scale. This is a scale that's used to score symptoms, and the score on the scale needs to be greater than or equal to 1. People with familial disease are eligible. People with sporadic disease are eligible. Really, the only group that's not eligible are those who have had radiation, probably as a child, and then developed CCM lesions where there's a reasonable correlation between the radiation and the development of those CCM lesions. And you can see here as well, those with treatment or surgery or bleed within a certain time period are also restricted from joining the study. So even though we have completed the phase 1 studies, again, those were studies that were done in healthy adult volunteers. The Sycamore study is the first time that we are administering REC-994 to people with CCM. And so because of this, and also the fact I'll talk about a little bit in a moment, that there are no validated scales for clinical studies for patients with or people with CCM, our primary objective of this study is to evaluate the safety and tolerability of the 2 doses of REC-994 in people with CCM. And you can see that this includes here, frequency of visits from every visit to every 3 months, the assessments on the left here. So we're looking at things like blood and urine tests, ECGs, where we're looking at the rhythm of the heart, taking vital sign measurements, physical and neurological examinations, and an ongoing assessment of adverse events that you would report to your study doctor. Even though there are no standard tools for CCM clinical studies, we actually have a number of efficacy assessments that are included in the study as secondary outcomes. And this includes looking at your lesions with MRI. We're going to be looking at the number and size of those lesions and how they change over time, as well as any evidence of bleeding that might be happening. We have some scales really that are mostly applicable to a stroke population, but they can give an indication of symptoms, and these will be completed by the study doctor. That's the modified Rankin and the NIH symptom score. We have what are known as patient-reported outcomes, or PROs, and these are intended to capture how the treatment affects your symptoms. Some of these are not specific to CCM, like the symptom screen or the SMSS and the PROMIS 29. And some of these are specific to CCM. And I do want to point out that we are including the CCM Health Index in this study as an outcome measure. This is a tool that's being developed specifically in this non-interventional study of the CCM high. In addition, we will look at the disease severity and measures of improvement. And these are qualitative assessments. These are done by the study doctor and also done by the study participants. And so these are what's called the clinical global impression, done by the study doctor, the patient global impression, which is done by the study participants, We will be using a diary, an e-diary, to capture information about seizures and headaches in participants who might be experiencing that. And we also, of course, as I mentioned a couple of times, we'll be looking at pharmacokinetics to see if people with CCM process the study drug differently than people without CCM. So that does seem like a lot. And this slide here is really meant to give you an idea of how long these assessments might take to complete, how long your study visits might be if you were to choose to join the study. So on days that include all the assessments, this can be about 45 minutes to an hour and 15 minutes, on the long side. This is, again, just the efficacy assessments. You would still have the safety assessments that would still need to be completed. On those visits that only have a few assessments, this could be as short as 5 minutes. So, you can get a sense from the slide what the burden might be for participating. Now, the study is open. It's up and running. And I want to give you a sense of where we are with the study. We are targeting 16 sites in the United States. We currently have 9 of those sites open. Those are the boxes that are in green. So that's Stanford and UCLA in California. We have a site in Phoenix. We have UT Southwestern in Dallas. We have several sites on the East Coast at Pitt, UPenn, Valley Hospital in New Jersey, University of Virginia, and Lyerly Neuroscience in Jacksonville. We have 3 sites that are close to being open. These are the ones that are in yellow, in blue, University of Rochester, Columbia in New York, and Jefferson in Philadelphia. And then we have 3 sites that we will hopefully be opening soon, Beth Israel in Boston, Emory University, and the University of Florida. So exciting, we were excited to see all these sites opening and patients starting to join the study. Now, noting that with 16 sites planned, we don't have full coverage of the country. So we do have some support systems in place that can allow participation for those who may not be close to a site. So Recursion, we will cover travel and hotel accommodations and arrangements. We have stipends in place. So if you have to miss work or be away for a little while, we also have a concierge service in place that can help to remove some of that burden for planning travel, planning study visits. Really, if you're interested, if your family members are interested, we want to remove as many barriers as possible for joining the study. Again, I really want to thank you for the opportunity to meet with you, provide some information on the program and the Sycamore study. If you want some more information, there are 2 sources that are available. Of course, there's clinicaltrials.gov. And we also have a dedicated website, www.sycamoreccm.com. Thanks again. And with that, I am happy to take any questions. I guess kind of went a little bit fast, but gives us more time for questions.
Oh, this is great. If you'd like to stop sharing, then we can see each other.
Absolutely. There we go.
Great.
Excellent.
Yeah, we have questions. So we're going to start. Well, I'll start asking them. The first one is actually sort of a side question. How did you come up with the name Sycamore?
Very interesting. Well, we had a competition within the company. We had a theme for trees. We have some folks in the company who are environmentalists, so to speak, and so for all of our clinical programs, we have a tree theme, and employees submitted their suggestions, and Sycamore was the one that won. Typically you'll see an effort to try and pick letters and put them into the study, but we really just wanted something that people would be able to remember and catch on to.
But it does work. The SY.
It does. Yeah.
The CA is cavernous. The M
yes.
is for malformation. I forgot what O is, but there's
Absolutely.
a thing.
It can work. It can work. Yeah.
Yeah. So, and I think it was shared with me that there's something about how sycamores were used in canoes, in birch bark canoes, to something from inside the Sycamore was used to make them watertight.
Yeah, interesting. I did not know that.
Yes, I did not know that either. And I don't know if you'll find that if you Google, that was just a rumor that was shared with me. It's a completely false story. All right. So someone else has asked, and this is again to sort of repeat yourself, but to really simplify it in layman's terms, what is the drug REC-994? And they're also asking if you could share any known side effects at this point.
Absolutely. So in layman's terms, it is, I don't know how lay this is, a reactive oxygen species scavenger. So essentially, it's similar to an inflammatory process. You have a buildup of these reactive oxygen species that can cause various problems. One of the well-known reactive oxygen species scavengers is vitamin D, actually, and so vitamin D, vitamin E, some of those vitamins, do a similar type of thing. REC-994 is a little bit more engineered in that sense, but it really is meant to mop up extra buildup of these nitric oxide compounds that accumulate within cells.
Okay, and then the second question was any known side effects at this point?
Yeah, so as I mentioned, side effects were pretty benign in our phase 1 studies. Sort of standard things like nausea and headache, which you see a lot of in phase 1 studies, but there have really not been anything notable to date from a side effect perspective. I do want to mention though that if there is interest in joining the study, one of the things that you will receive before joining the study is what we call an informed consent form. And the informed consent form really lays out the risks for not just receiving the study drug, but for some of the procedures as well. So you have an opportunity to read through that, ask questions of your study doctor, and really make a determination. If these are risks that are essentially the risk benefit, personal risk benefit for participating.
And I think too, from understanding the structure of the trial, having so many visits, as much as it feels like it might be a burden, it also gives an opportunity for oversight in
Absolutely
intense ways. We know of a different trial that was run where people only had to be seen every 6 months or even once a year. Being seen every 1 to 3 months means that if anything is going wrong, someone can be right on top of it.
Absolutely.
Okay, so a few people have asked, when REC-994 is proven to work and approved, is this a medicine that someone would need to stay on for the rest of their life, or is it something that you would discontinue after a period?
That's an interesting question. I think it's one that we will continue to evaluate. Our preclinical data does suggest that this would be, a lifelong treatment. But there's an opportunity for emerging data, certainly that could suggest that there's opportunity for us to study this a little bit further. Let me put it that way, to determine whether there's something that you would stay on, or after a certain amount of time you discontinue and still reap the benefits.
Okay. From the mouse studies, can you speculate? Probably not, but I'll ask it anyway. How soon do you expect to see improvement in cell permeability?
Mm-hmm. So extrapolating from mice to animals, of course, is challenging. The mice studies are certainly shorter in duration. We are looking in the clinical study at efficacy measures, MRIs at 6 months and at 12 months. And so, we have some expectation that at 6 months, we might see some evidence for a difference between the active drug and placebo, certainly more at 12 months. But we also, I think a large part of the extension study that I mentioned is also an opportunity to look at longer time points as well. Do we see an improvement that's continuing to emerge over time, or do we see an improvement early, let's say at 6 months, that then just remains stable?
Okay. In terms of qualifying for the study or also, let me ask this in two different ways. First
Sure.
is in the mice, was REC-994 tried in all three genetic versions of mice, CCM1, 2, and 3 before going to trials?
So we have some evidence internally that in all three mutations in the publication, the main publication, it's CCM2 is the main publication, is the main mutation in the publication.
Okay. And then for enrollment, is there any restriction on which mutation? Can you be sporadic? Can you have CCM1, 2, or 3, or is it limited to anybody?
It's all of the above.
Okay.
Any familial, sporadic, any of the mutations.
All right. Someone wants to know, is there any chance that Salt Lake City could become a study site?
We would certainly love it. We're doing whatever we can there, but as of now, no.
Okay. I'm not sure you're allowed to answer this, but how many people are currently enrolled? Are you allowed to give that
I will
answer?
answer in vague terms. Let's say patients or participants are enrolled. Things are moving along well. We're still in the early days, but I cannot give a specific number.
Okay, I figured that. Someone is asking that, or is sharing that they just had surgery on the 1st of September due to a bleeding lesion. Can that person assume that they would be ineligible for this phase of the study? So that would have been 3 weeks ago. Or
3 weeks ago. For now, yes. However, I just want to make sure I'm not misquoting. Potentially in 3 months since the bleed, since the surgery, you might be eligible. So 3 months for the bleed. Potentially, if you had brain surgery, it might be 6 months before eligibility to start screening.
Yeah, and they had surgery, and so,
Okay.
but a couple of caveats just to make sure people understand. You have to still have a lesion. So you still have to have a lesion, and you have to have a lesion that has symptoms. So if you had your one lesion removed in early September, you're probably not going to be eligible. And if you have multiple lesions, but that was the only symptomatic one, then you're probably not eligible at this point. All right. Someone is asking, they might have missed how long is the blinded placebo control part of the trial versus the open access comes after?
Yeah, absolutely. The first part of the trial is 12 months.
Okay. Someone else is asking, and this, I'm on Coumadin, will that disqualify me from participating in the study?
No, based on the inclusion exclusion criteria for the study, that would not be exclusionary. But certainly that is something that the study doctor can further evaluate.
Okay, and I don't know, I haven't scrolled down far enough. There's still a ton of questions, so yay. But the question has been asked of me multiple times. I take atorvastatin for cholesterol.
Mm-hmm.
Am I disqualified because I'm on atorvastatin or propranolol?
Right. The answer is no there. Similar with Coumadin, with the statins, we would be looking for the dose to remain constant during participation in the study. Certainly, we know there obviously is a statin study, and of course, people are on statins for heart disease, and of course. So that is not exclusionary. It would just need to remain as static as possible during participation in the study.
Okay. And then a follow-on question is, how does this medicine work differently from medicines that are already in trials or have been in trials like atorvastatin and propranolol? Is there a simple way to answer that question?
So I think I can answer the question for REC-994. So as we talked about the mechanism of action being reactivated oxygen species, which is one axis of mechanism of the disease. There's also another pathway that runs through something called ROC kinase, where we also think that we have some effect. This is still hypothetical, we think we have some effect there as well, as opposed to atorvastatin, which is really working on one axis of the disease.
Okay, all right. The MRIs being done in the study, will they be with or without contrast?
They're without contrast.
Yes. And they're very short. They're like 25 minutes long. How does an FDA-approved drug go on to approval for pediatrics?
That's an interesting question. I'll see if I can give a short answer. There is a prescribed pathway. Essentially, we would have to run studies in pediatrics to gain approval, and we need to work with the FDA to understand the dose for pediatric patients, whether it's going to be a lower dose, understand the pharmacodynamics again, what the drug does to the body, what the body does to the drug. Is that different in children versus adults? So there would certainly be work that needs to be done both from a clinical study perspective, but also perhaps some preclinical work. We'd have to look at some what we call juvenile toxicity studies. So we'd have to, for example, in rats or mice, give the drug to juvenile animals and get a sense of the toxicities that may be the same or different in the juvenile and from the adult animals. So there's certainly some work that would need to be done.
Okay, all right. I know a lot of our families have affected children and are just dying for something.
Absolutely.
Okay, so if someone joins the extension study, how long does that last? Is there an end to that?
The extension study right now is 1 year. There's a reasonable expectation that if development of REC-994 is continuing, that we would look to extend that extension study, but it's 1 year at this point.
Okay. All right. A specific site question: which Columbia University is the study at, and when does it begin, and how long will it last? They want to know basically how to get into the Columbia site.
I'm actually asked you to handle that one.
Okay, so it's Columbia Presbyterian, and the PI there is Dr. Sandra Connolly, who's in the neurosurgery department. Our website at this point already has, and so does clinicaltrials.gov, has the contact information. I believe Dr. Connolly right now is just reaching out to his own patients. We have not yet been asked to broadly disseminate the fact that they have the trial there. But if you're already a patient at Columbia Presbyterian, you can expect to probably get contacted, or you can just go ahead and proactively contact them. It's begun. So call them, tell them you want to be in there, get your, if you're not already a patient, they'll want to see some records of yours to make sure that you're, a potential candidate, and then they'll bring you in for a screening. And as Dr. Morrison mentioned, on the screening day is the day that you get your MRI and you fill out all kinds of forms and you get blood work, and all of the things happen to make sure that you really are appropriate for the study. And then you, if you are, you take off from there. Did I get that right? All right.
Absolutely. Yeah.
All right. Is there a certain number of people you need in the study to properly evaluate?
There is, and that's the 60. So when we design the study, we look at some statistical elements and ensure that we included enough people to make sense of the outcomes of the study.
Okay. How can seizures or headaches be considered symptoms to be enrolled in the study?
Yes, but again, this will be determined by the study doctor. But yes, those absolutely are symptoms, relevant symptoms.
But they're not. Can they be the only symptom? Can someone just come with a headache?
If it's reasonably expected that it's due to the lesion, that will be allowed. Yeah.
All right. Do you need to have had a symptomatic lesion within the past 18 months to participate, or just any lesions that were symptomatic in the past, as in years ago?
Interesting question. So you have to have symptoms now, essentially, to be eligible for the study.
Yeah, they And the reason for that is because they need to see if the drug is having an effect. And if you don't have
Absolutely.
to start with, then they don't know what's happened other than what they're seeing on imaging. None of the survey data, will be relevant. But I think, and you can correct me if I'm wrong here, if the medicine is approved, if it gets to that point, then the criteria for who can take it will probably be different, and may indeed include people who are using it for prevention and not just for treatment. All right. When folks are in the study, do they receive copies of the MRI reports for the MRIs that are done in the study?
There's no restriction per se. So I would say yes, but there might be a timing element there. And so this would be something certainly to discuss with the study doctor. We likely would restrict that, unless it was for care from your primary physician, for example, until after the study is completed. And then you would be able to get access to those. But during the study, it's unlikely that you would get access to those images during the study.
And again, I will expand on your answer. The study's blinded. You're not supposed to know if you're on the medicine or if you're not on the medicine or what dose of the medicine you're on. There's three there. You could be on 200 milligrams, you could be on 400 milligrams, or you could be on placebo. If, by, some miraculously, all of a sudden your MRI looks better, that might give away what you're taking, and you're not supposed to know that. Study doctor's not supposed to know that. So, when did this person is asking how long has this specific clinical trial been around? But I think the better way to ask that is when did the phase 2, the Sycamore trial open?
It opened in, I believe it was earlier this year. It was in March. Was the first enrolled participant.
Okay. I'm sorry. I have some longer questions that people have put in we're almost to the end. So y'all, if you want to ask a question, now would be the time to start typing. And also on my team, if someone could drop the link to the support group in the chat now, that this would be the right time to do that. So one more time, someone is asking, I feel like we need another layman's explanation of how this is expected to work. Does it shrink? How does it shrink lesions, prevent new ones, affect other unrelated, and does it affect other unrelated body functions or organs?
So how does it essentially it improves the cell wall integrity, and so if the hypothesis being that because of the cell wall integrity, there is these growth of these lesions because of abnormal growth and clumping of cells. What REC-994 can do is, through disrupting the buildup of reactive oxygen species, improve the integrity of those cells so that they're more normalized. And the thinking there is that if cells are more normalized, then the vasculature can also be restored. And so then the size of the lesions that are due to the abnormal vasculature would reduce, or ultimately also would not form in the first place. Does that help?
I hope so. The question then was, would you expect any impact on other body organs, body functions?
So interestingly, the impact of CCM seems to be localized largely to the central nervous system, to the brain, which is where our action is happening. So we don't believe that, and this is something we'll evaluate during the study, of course. But our animal data doesn't suggest that we would have effects outside of the brain. But this is something that we'll be looking at.
Okay. All of a sudden, more questions are rolling in, people realizing it's near the end. All right, someone has asked, is this a repurposed drug? And if so, what other uses has it been evaluated for?
It is a drug that has been out there before. It's been used in radiation, post-radiation exposure as a topical. So it's, it is a drug that has been out there.
Okay. Will there be a lapse between the end of a person's participation in the study and the beginning of the extension for enrolled participants? Do they have to wait for everybody to finish?
No, great question. Once you finish the 12 months in the double-blind study, you would move right into the open label study.
Okay. I have seizures due to my cavernoma. If the seizures stop or get worse, how would you know whether this was because of the medicine or just would have happened anyway?
Mm-hmm. Yeah, so we may not know 100% for sure, but the way we try to get to this is during the screening period, you will have your diary and we'll be recording the frequency of seizures and headaches. And when you start taking the study drug, whether it be active drug or placebo, we'll continue to monitor the frequency. And so we'll be looking to see if there's a change in the frequency of seizures or headaches before starting the drug and after starting the drug, and look to make correlations in that way. And also recording of other symptoms as well.
Yeah. And they have to have enough people participating to be able to find out. That's a big part of it. It's just making sure an N of 1 is not enough. It's
correct.
Many people. There's a potentially last question, and it's on the technology side, just differentiating Recursion's AI platform from CRISPR. Everybody's heard of CRISPR-Cas9. I know that they are hugely different, but they get confused in the minds of our families sometimes. Can you share the difference?
So those are technologies that we use within our platform. So in, again, very broad strokes, what our platform aims to do is to introduce perturbations into cells. And then we can look at how that changes the phenotype of the cells. And then we look for compounds that can reverse that phenotype. And one of the tools that we do use to introduce those perturbations is CRISPR. So it's something that we definitely leverage.
Okay. So the answer is CRISPR gets used within the broader spectrum.
Correct.
You all do.
Yeah.
All right.
But it's not our only tool.
Okay. As far as I can tell, that is the end of the question. So thank you so much. This has been so informational and helpful. I would like to encourage anyone who has extra questions, you can send them to me. I will pass them on to Dr. Morrison, or unless I can answer them myself, and then I'll try to answer them. This is our first industry-sponsored trial. Is really, really important that we support it, that we come out and we show that we want treatments, and the only way to get treatments is to participate in the trials. So I know it's hard. It's putting yourself out there. It's taking a risk, but I think in the end, it's incredibly worth it to be part of this process. So
it is, and we're extremely grateful. I mean, it takes a lot for individuals to want to participate in trials, and we certainly appreciate the dedication of families willing to participate.
Yeah. Okay. Well, thank you.
Thank you.
And for everyone, we're going to meet again next month. Dr. Awad will be here. He'll be talking about CCM and the aging brain, he and a few members of his team. So we hope to see you back then. In the meantime, go click on that link and join the support group and talk some more about REC-994 and the Sycamore trial. So,
excellent.
Thanks.
Thank you, Connie. Thank you, everybody.
Good night.
Bye-bye.